आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Indian Statistical Institute is a public university which is recognized as an institute of national importance. It grew out of the statistical laboratory set up by Prashanta Chandra Mohananobish in Presidency College, Kolkata. Established in 1931, this unique institution of India is one of the oldest institutions focused on statistics. Primary activities of ISI are research and training of statistics, development of theoretical statistics, and its applications in various natural and social sciences. In this podcast, we are immensely happy. We welcome Professor Sanghamitra Bandyopadhyay, the first woman director of Indian Statistical Institute. Dr. Bandyopadhyay shares about her fascinating journey in the world of science and research. Dr. Bandyopadhyay shares her experience from a passionate researcher's perspective, as well as from the perspective of an efficient administrator.
Thank you very much. Well, you see, I grew up outside Bengal till about my 9th standard, after which my father got transferred to a place where there was no good school. So I had to move to somewhere close to Kolkata in Howrah district. I stayed at my maternal grandparents' home. And from there, I continued my studies past class 10, 12 from Bethune College. And then because I was in love with mathematics and physics, I got an admission in engineering, but then I also got admission in Presidency College for physics, BSc physics, which at that time, there was a craze for studying physics, and I took that. Completed my physics, but it became so theoretical that I realized that I was more of a hands-on person. So at that time, Calcutta University had this provision of moving from physics or mathematics, BSc, to BTech in computer science. And that was a 3-year BTech program. I moved to computer science. And that move turned out to be extremely good for me because I liked and loved computer science. And then I stayed on, of course. I did my BTech. After completing BTech in computer science from Calcutta University, I moved to IIT Kharagpur for my MTech, did pretty well. And then came back to Kolkata to Indian Statistical Institute to complete my PhD from here in 1998. I submitted my thesis and defended my thesis. And then I went for a postdoc to Australia. I also was offered a position as a lecturer in Indian Statistical Institute, which I took. I think very few people are able to continue living close to their homes and also join an institute as reputable as Indian Statistical Institute. So I was very fortunate. I continued as from lecturer, then became assistant professor, associate professor, professor. So it went with time. Of course, research was my first love. It's extremely difficult as well as fulfilling to think of something or to come up with something which is novel. And that's what research is about. So, and then I was selected as the director of the institute in 2015. I happen to be, of course, the first woman director of this institute, which this institute is a 90-plus-year-old institution. And also, I was quite young at that time, so I burnt my fingers, actually, in administration. And thereafter, in 2020, when my first tenure ended, somehow I got selected again for a second tenure. So, and that after Mohananab is the founder of this institute, nobody continued for a second tenure, which I did. And so that was also sort of first in this institution. I continued doing research, and right now also I continue doing research, though a lot of time is spent on administration. But my research has been generally in the area of artificial intelligence, machine learning. In particular, I was always interested in optimization. You see, we are always optimizing our time, our money. We are always going through this process of optimization in our daily lives. And many of these problems, if you think about it, are multi-objective in nature, in the sense that there are multiple objectives which you want to optimize at the same time, while these objectives conflict with each other. You want to make one objective good, the other becomes bad. So how do you handle such problems? So a simple example could be, you are planning a trip from point X to point Y, and you want to bring down the time, you want to bring down the cost. And you can imagine that bringing both of them down, minimum time, minimum cost, that doesn't happen together. If you bring down the time, the cost will go up. If you bring down the cost, the time will go up. So how do you optimize under such situations? This is a very simple example which I gave, which there are many, many problems in science where the problems are much more complicated, much more complex. The objective functions are not just two, they may be many. How do you solve such problems? A typical way of solving these problems has been that you just join the objectives together. So suppose you have two different time and cost. You, give a weighting factor to time, give a weighting factor to cost, add them up, and then you have one number, optimize that. Because single objective optimization, there are many techniques which work fine there. Now, the problem is, it's often not a good idea to join them together. It is much better if you can actually keep them separate, optimize them simultaneously, not one after the other, simultaneously. And at the end, what you will get is a set of solutions. You will not get a single solution here. Because when you have a single objective, then you look at solutions, you try to improve, come to the best. But that's only a single solution. But when you are doing multiple objective optimization, you are actually looking at many solutions which are trade-off. In one objective is better, in another objective is better, and so on. So you're looking at a trade-off surface. You're looking at a set of solutions, and together you try to improve all these solutions. So these actually have a lot of applications. In particular, we've applied it to many problems in biology. One important problem where we applied it to was in the area of drug design. So drug design, as you can imagine, drugs are small molecules. Okay, these small molecules, when we ingest a drug, what happens is the small drug molecules, they go and find their targets inside the cells. And these targets are other molecules within our system, but these are very often proteins. Okay, proteins, these are not the proteins that we say, okay, eat that or eat this, it has protein. These are not those proteins. These are proteins which are created inside the cells, okay, inside our body, inside the cells of that make up our body. So those proteins, either one of my own protein, the host's protein is misbehaving, something has happened because of which some wrong functionality is becoming apparent and the disease condition is appearing. So you have to do something about that protein which is in the disease pathway. Or it could be a pathogen which has infected you, a virus or a bacteria, and there are certain proteins which are important for the survival of the bacteria or the virus. You may want to, through the drug molecules, target those proteins. So whatever it is, more often you have for drug molecules, there are protein targets. So you have to design those drugs using the proteins as the target. And not everywhere in the protein is, not every location of the protein is important. Because protein is a three-dimensional molecule. Just think of it as a three-dimensional structure with certain clefts and cavities on its surface. And it is these clefts and cavities, many of those cavities are actually where other things come and bind, sit together with the proteins, and that is how the protein manifests its functionality. Now, if that function is going wrong, something has happened and you want to actually put a drug molecule into that cavity, even before the harmful binding takes place, the drug molecule is already sitting there. So if the drug molecule is sitting there, then that harmful binding hopefully will not take place. And if that harmful binding does not take place, you have some hope of curing the disease or doing something about that disease. So that is the basic philosophy of most of the drug design activities. So typically drugs were discovered by chance, by chance observations. But that had a very, very high chance of failure. Now, then came, scientists now had millions of molecules in their database and they had a target. They tried every molecule. So this was like a screening which happened. Most of the molecules ended up as failures, did not work. But that was very time consuming and costly. So then came computational methods where you just don't blindly try out the target and the molecule, but you use logic, that this molecule is likely to act, this molecule is highly unlikely to be active. And therefore, without even actually doing the testing, you discard many molecules because they may have some electrochemical properties. Suppose you know that a negative charge and a negative charge would repel. Now, suppose that the protein has a negative charge on it, and the drug molecule that you have that also has some negative charge. So then these will never sit together. That's very common sense. So accordingly, many such other principles were used, and that developed the area of rational drug design. So rational drug design, there were some success stories already. We also tackled this problem of rational drug design. We tried to design small molecules, assuming that the target protein is known. And not only is the target protein known, but its active site, the site where the harmful binding is happening and where we want the drug molecule to go and sit. That active site, that functional site of the protein, target protein, is known. By known, I mean that every atom making up that area, the coordinates of every atom, whether it's a carbon or a nitrogen or an oxygen or a hydrogen, the coordinates are also known. And that is possible. There are databases which tell us all these things. For example, there's the protein data bank, which gives us the coordinate information. So what we did was, we were designing small molecules which would sit in the active site of the target, and we realized that this is actually a multi-objective optimization problem, because we know that in physical systems, anything with a low energy is stable. So the molecule that we are designing, if that molecule has to be a stable molecule, the drug molecule has to be stable. So if that molecule has to be stable, it must be associated with low energy values. If the interaction of this molecule with its target has to be stable, then the interaction energy has to be low. So we are already trying to optimize two objectives. You can see one is its own energy and one is the interaction energy. Then there are other energy components which together have to be minimized. And there are other components, not just energy, but other objectives which we have to optimize. For example, you might design a nice molecule which fits very well in the functional site of the target protein, but the molecule itself is so heavy and big that it doesn't enter the cellular membrane. It cannot go inside the cell. So if it cannot go inside the cell, it will not find its target. Target is inside the cell. So there are other measures by which you can actually quantify these. That's called oral bioavailability measure. So there were three different objectives which one is the energy component, one is something called Tanimoto coefficient, and another is the oral bioavailability measure. Plus, this is a constrained optimization in the sense that not every molecule, even if it is good, there are some other properties that this molecule must satisfy in order for that molecule to behave as a drug molecule. And these are called ADMET properties. One of those, for example, is toxicity. This molecule may be very nicely fitting here, but if the molecule itself is toxic, then you cannot use it as a drug. So there are some ADMET, that T is for toxicity. So this is a constrained multi-objective optimization problem. And we had developed in another one of our journal papers, one multi-objective optimization technique, which in our lab with my students I had already developed. We used that technique for this drug design problem. And we used as the target a bacteria protein of Mycobacterium tuberculosis. It's a REC-A protein. The name of the protein is REC-A. And what the protein does, when bacteria has infected the human body, then we take antibiotics. The antibiotics often damages the bacterial DNA. REC-A protein, which is of the bacteria, it repairs that DNA and hence plays a major role in survival of the bacterium in spite of the antibiotic. And so what we did was, if we target REC-A, then we hope that the bacterial DNA will not be repaired. So we used that as a target and we designed certain molecules. Our method evolved certain molecules, of which a few were already known to be inhibitors of REC-A protein, because we were not working with REC-A for the first time. People had worked already. So there were a few molecules which were already known to be inhibitors. So that gave us confidence on our method that probably it's doing something right. But there were, in addition to those known molecules, we also designed some new molecules. And then we did further testing on the computer to study its activity structure, activity relationship, etc. After which we published, because after that our expertise ended because we were unable to really take the research further. But our computational method we published, and our hope is somebody somewhere takes it up. So that's one of the things which I have done. But also, you see, when I was young, in my school days, while I was a fan of physics and mathematics and languages, I was really, really afraid of biology. And the first opportunity I got of leaving biology, I left it. But the thing is, when I was a young faculty, I had a student who had a biology background, and he convinced me that the clustering and classification methods, we were developing all these methods I did it for my PhD as well as postdoc studies. So the methods that we were developing had a lot of applications in biology. So what happened is that student taught me biology and I taught him computer science. And that went on for 1 to 2 years because it was not easy for me to remember all those biological terms. Every day he would teach and I would forget. So, but thereafter, gradually it stuck. And with that student, actually, we worked on a small molecule, computationally though, not in the biological wet labs, but on computers. We worked with, a small molecule called microRNA. At that time, those were the initial days of microRNA research, particularly computational research on microRNAs. And these microRNAs are very interesting molecules. These are small RNA molecules, okay? So we know DNA, our genes are actually DNA, genetic materials is DNA. So certain portions of the DNA strands are genes. Now the DNA, through a process called transcription, makes RNA, and RNA through a process of translation makes proteins inside the cell. This and these two steps together, it's called the central dogma of molecular biology. And anybody doing any research in molecular biology, this is something that a person must know in detail. Now, the RNA molecules, which make proteins, those are called messenger RNA. But there are many genes which make RNA, which are not messenger RNAs. For example, microRNA is one such RNA, which makes the genes of the microRNA genes make small RNA molecules, which are called microRNAs. And the task of this microRNA is to go and interfere with other messenger RNAs. Remember that messenger RNA, if that microRNA was not there, then the messenger RNA normally would have produced its protein. And let's say the protein that the messenger RNA would have produced helps in cell killing. Let's say, it's an example. Now what happens is cells are dividing and therefore cells also need to be killed. And there are proteins which are involved in that cell killing. Those are called, let's say, apoptotic proteins. Now a microRNA, because at certain point of time, because of certain genetic reasons or environmental reasons or whatever, the microRNA gene, which was sitting in the genome of that person, but was sitting silently, becomes active at some point of time in the life. And then this microRNA gets produced, and this microRNA does what? It goes and either degrades the messenger RNA or just binds with it, but the effect is the same. The messenger RNA is no longer able to produce its protein. Now this protein, when it was produced, what was it doing? It was killing cells. Now cells are dividing, but because the protein is not getting produced, cells are not being killed. And that means an explosion of cells in a particular region of the body. And that's cancer. MicroRNAs were found to be involved in almost all types of cancer which were there, plus many diseases. So studying microRNAs was very important. And we were sort of the first movers in that direction, that student and I. And we did, plus other students joined. And we did a series of works on microRNA. And that caught a lot of international attention because we, for the first time, said that a microRNA directly inhibits or stops a messenger RNA, another mRNA, its target molecule. But there are secondary effects of microRNAs, which actually in a loop, it can actually also make another messenger RNA active. So it's not only degrading, but it has secondary effects. And that was not really studied at that time. Nobody was thinking in that direction. So that became a sort of firsts. And that attracted a lot of attention. I got international invitations, national invitations as well to go and speak. And that became very important. And then we took this study further. We studied microRNA involvement in, let's say, breast cancer and colorectal cancer. And through our methods, which can be called graph theoretic methods, because we were doing analysis in the level of graphs, we came up with some new genetic biomarkers. So microRNA markers. We were able to identify, for example, 2 microRNAs in breast cancer about which not much information was available at that time in the literature. And about 2 years after that study was published by us, one of those microRNAs was actually validated in the lab by a group absolutely independently. So those are the things which actually give us a lot of pleasure in the sense that what we are predicting computationally is turning out often to be true, to be correct. So essentially what we do, you see the statistical people from statistics background or people like us from computer science background, when we are working in the biological domain, you must realize that this is not the end of it. It is just a tool. We are using it as a tool because the biologist or the biochemist, they are actually searching for a needle in a very big haystack. They don't know what to look for. They are just, grappling there in the dark. For us, through our methods, the better the method is, better it is. What we are doing is we are reducing the size of that haystack so that the biologist's life is simpler. Because anyway, their life is very difficult because the possibilities are immense. So many possibilities are there. So if you can come out with certain predictions and high confidence predictions, or if you can with high confidence say that this is not going to work, no point in trying it out. That is a big thing for the biologist. So that is what I have been doing for so long. And I consider myself quite fortunate to have got an opportunity to do my PhD in this institution. So the Indian Statistical Institute, it was founded by Professor Prashant Chandra Mahalanobis in 1931. Mahalanobis was a physicist by training and he taught physics in Presidency College till he superannuated. But it was a chance introduction to statistics when he missed his boat from England to India during the First World War. And he got hold of certain journal, one journal called Biometrica, which he read the papers while coming back to the country, to India. And he realized the importance of statistics in nation building, for human welfare, for social development. And with like-minded people, he started Indian Statistical Institute in Presidency College, in one small laboratory in Presidency College in 1931, 17th December. And at that time, they started with a 200 rupees budget. It's now grown many, many folds. Now, ISI, with headquarters in Kolkata, has centers in Delhi, Bangalore, Chennai, and Tezpur. We have a branch in Giridih. We have a campus, a unit of the Statistical Quality Control and Operations Research Division in Hyderabad, and very small units of SQC and OR in Mumbai and Pune. We have some land also in Baroda. And this institute has 7 scientific divisions: the Theoretical Statistics and Mathematics Division, Applied Statistics Division, Computer and Communication Sciences Division, Social Sciences Division, Physics and Earth Sciences Division, Biological Sciences Division, and Statistical Quality Control and Operations Research Division. So this institute actually, after Mahalanobis, we had C.R. Rao as the director of the institute. He passed away very recently in August, but a legendary figure. And thereafter, the institute has nurtured and produced many, many outstanding scientists, not of course in statistics and mathematics, but also in many other domains, including economics, computer science, and social sciences in many areas. Mahalanobis started a flagship program of the institute, BStat and MStat, in the 1960s with JBS Haldane. The design of that BStat program was so unique, it remains relevant till today. And today, when India is moving towards general education, where a person with mathematics background has to study biology, and a person with biology honors has to study mathematics, etc., what India is doing now, I cited it in 1960s, when the statistics students had to study biology, physics, economics, social sciences, linguistics, psychology, what not, because these were the domains from where data came. Geology, if you see, we have a geology museum here, a fully mounted dinosaur fossil, only one of 2 in the country. The first one in ISI, the second one is in Hyderabad in Birla Museum, which was mounted with the help of ISI scientists. So this is a unique institution. Anywhere around the world, if you see a statistics department and Indian name, more often than not, that person will have a connect with Indian Statistical Institute. So it's a proud legacy which we have, and we struggle every day to meet up to those standards. But we do try. We have outstanding scientists in the institute at this point of time also, and we hope to keep on producing outstanding students and outstanding researchers. Thank you very much.
We feel enriched to listen to Professor Bandyopadhyay in this episode. We will remain ever indebted to her for her competent leadership and inspirational vision. We thank her once again for such a beautiful deliberation.